All right, it is time to get started. Welcome, everybody, to the U.S. Territories Wastewater Operator Training Series. This is session 13 on Activated Sludge, Part 2. And my name is AJ, and I'll be your trainer today. So just a little bit about me. I am a research engineer with the Southwest Environmental Finance Center, and we will be moderated today by Valeria Cortez Mora, our professional intern. If you're new to us today and just joining us, we are the Southwest Environmental Finance Center, and we are out of the University of New Mexico School of Engineering. And this training series is funded by the EPA. A couple quick reminders. You can use your toolbar at the bottom to write in questions to the chat, to unmute yourself or enter your video. Please feel free to enter questions in the chat throughout the training, and Valeria will let me know, and I'll go ahead and try to answer them the best I can, or just in whatever order they come in. So about this training, this series is designed to supplement information based on these WPI and ABC operator certification for our operator certification classes. Our training kind of mirrors a lot of the information that's in there and is designed to help you guys kind of pass those tests, as well as from an operator point of view of how to operate different types of facilities. Here's the schedule so far that we've had on all the different topics we've trained on. We are here in this list for the year. So this is the list of all the trainings that we've had this year. And then we will continue again in the new year and start that back up, I believe, at the end of January. If you want any information about our previous trainings, those can be, the training recordings and copies of the slides can be found at our website in the Cert trainings folder. And you can see all our past recordings, like I said, and get copies of this information to help you guys kind of study or as reference material. Now, the last part is just be sure that everyone attending your group sign the sign-in sheet, so we can make sure to track that and make sure you get your credit hours for the sessions. With that, we can go ahead and start our training today. So like I said, this is Activated Sludge. This is part 2 in the series. And the first thing we're going to talk about is Activated Sludge, characteristics and controls. If you remember from last week, those of us who joined us, we discussed kind of an overview of the activated sludge process, its role in wastewater treatment, and the components that go into it. And we'll go more in today about how to control the process and what to do with our different variables and how to implement our findings and our observations and our calculations. So that's where this training is going to really focus. Now, we did introduce some of these variables that we monitor as part of the instruction for our components, and then we'll kind of go into more details and how they fit in actual process control. So to start, I do want to quickly discuss why the characteristics and control measures of activated sludge is important. Activated sludge you'll sometimes see through here might be abbreviated as ASP. In a lot of the reference material, it is referenced as ASP as well. But ASP, just like I said, references activated sludge process. So last time we talked, we mentioned that activated sludge processes are biological processes and have more variables than our kind of physical processes, or this is important because there are more process variables than our typical physical processes, such as or chemical processes such as sedimentation and screening, or disaffection and coagulation. Activated sludge is a more delicate and sensitive process than our physical processes. So operational data directly in the process must be collected as well as data upstream and downstream to evaluate and make changes to the activated sludge process. Now, with our activated sludge process, because it is a biological process, the characteristics change daily. and the sludge process objectives must change with them and be maintained in a balanced state to account for any minor changes that could throw it off or limit our treatment. Operators must also be ready for the unexpected and understand in detail how this specific process and their specific activated sludge process can be controlled to make up for any emergency change in characteristics or change in variables. So the objective of trying to control those variables and why we're actually monitoring our activated sludge characteristics, some of the backbone of that are to maintain the aeration tank to produce those flock-forming bacteria, to flocculate and settle the flocks into the secondary clarifier, and to manage the activated sludge to maintain a healthy proportion of microorganisms in the system, particularly in the aeration tank. So those are the goals that we're trying to accomplish by implementing process controls on the different variables such as DO and sludge age. So the first thing that, the first thing we kind of monitor in relation to our activated sludge to understand what process changes go into it are going to be our influence characteristics. So it is important to understand the physical and chemical makeup of the influent that will be treated by the activated sludge process. Analysis of our influent characteristics helps with treatment and making process control adjustments. As we mentioned, certain influent traits such as infiltration and inflow, high septic loading, and the different types of wastewater discharge from industries can have effects on our activated sludge process. For example, infiltration and inflow or I&I can result in excessive grit and silt that is washed out. It may damage process equipment such as pumps, mixers, and our aeration diffusers. Things like changes in influent flow, especially higher flow in the collection system, can result in the washout of organic buildup in the collection system that can increase the treatment requirements of the activated sludge process. Then I&I can also cause temperature changes in the influent. It's usually a result in a colder temperature of our influent due to things like rainwater that infiltrates the system and decreases the temperature of the water in the activated sludge process, which can inhibit our normal operation. Then, with sitting water in our wastewater, we can also have septic shock that can threaten our system as well. So we do have to be aware of what's going on in our influent and the characteristics of it in order to make adjustments to our activated sludge, such as increasing wasting, increasing holding times, increasing aeration, and stuff like that. Finally, it's also important to understand what types of industries may contribute to the collection system, and if they contact us, what kind of impact we should be ready for and how to adjust for that. And then sometimes this is done through the implementation of pre-treatment principles or even a whole pre-treatment program. Now, like I said, we're going to mention a lot of kind of parameters that we've mentioned before. Here are our influent parameters that we like to take into account. They include flow rate, total suspended solids, volatile suspended solids, biochemical oxygen demand, chemical oxygen demand, total kilojoule nitrogen, and ammonia and nitrate. We mentioned all of those last week and how they're kind of calculated. But now we're going to go into what kind of calculations we actually use them for to make process adjustments like wasting, like understanding our sludge age and stuff like that. I'm sorry, those are the influent parameters. Now, similarly, we also have our activated sludge processes, which we still mentioned. Things like our dissolved oxygen, food to microbe ratio, mixed liquor suspended solids, mixed liquor volatile suspended solids, solid retention time, mean cell residence time, and sludge volume index. We'll kind of discuss how to calculate all of these and what they mean for our activated sludge process, and why these are important for our process control and how the variables that we just talked about can help us understand them and what they tell us about our activated sludge process. Also in this training, we will talk about some common issues in our activated sludge. These are some of those common issues, and we'll go into more detail about them later, but they include bulking sludge, foaming, settling problems, over production of sludge, nutrient imbalance, organic overloading, and then poor flocculation. So with that, and before we kind of go into our more analytical views, we can go into a little bit more detail about our observable characteristics of activated sludge. So our healthy activated sludge has some pretty easily observable characteristics that operators can use as indicators to identify when problems may be developing in the activated sludge process. As with most processes, and you'll hear me reiterate this a lot, each plant will have their own kind of unique characteristics. So it's important for operators to understand what their plant is supposed to look like when it's running well. But many plants do kind of share these common observations, and this offers these characteristics I'm going to introduce right now offer a good starting point for what to look for, or may even offer some insight into why a wastewater plant isn't operating correctly. So the first characteristic I'll talk about are the visual ones. The first is color. So a lot of times your activated sludge should have a solid brown and sometimes grayish color activated sludge. You should have diffused air should be visible. A healthy DO level would be indicated by fine, crisp white bubbles, and this also will not be discolored bubbles, or you won't see an oily sheen. And then also in your settling tests or your settling samples in your secondary clarifiers, you should see the formation of well-structured and dense flocks of microorganisms that settle easily for a distinct and dense sludge blanket. So what we're saying there is you should, when you have a sample of activated sludge or your secondary clarifier, there should be a distinct difference between the treated separated water, which we call supernatant, and the settled sludge itself. Then activated sludge also has some distinct smells to it. And this tends to be an earthy, fresh soil smell, like fresh dirt or soil when it rains. Good, healthy activated sludge and an activated sludge treatment process that's running correctly should smell like that. Now, if you have foul odors, if it smells, if it has that rotten egg sulfide smell, that means that there may be some issues with your activated sludge process, such as extra sulfur. There could be chemicals that are inhibiting your process and stuff like that. And it could indicate that there are process updates or issues going on with your facility. So just based on kind of sight and smell, those are two things that kind of help us understand if our activated sludge process is running correctly. Now, to account for some of those issues, the first, that unusual coloration or visual observations, you want to ensure first that your aeration levels are correct. So DO concentration should be between 1 and 2 milligrams per liter to ensure fresh activated sludge. Unusual bubbling would indicate that there's a need to inspect the distribution lines for any breaks or leaks. So if you don't see kind of those crisp, small, clear bubbles, and you see giant bubbles all over the place or from one specific place, that may indicate that you're not getting aeration to the whole basin because there's a break in the line. Or if you have large bubbles throughout the process, that may indicate something chemically is going on, like there's an oxidizer in there somehow from an industrial source or some sort of chemical from domestic sources and stuff like that. So, if you're not seeing the typical clear bubbles, white bubbles, if you're seeing big giant bubbles, that may be an indicator of that. Now, if you start to have unusual smells, that could be from your sludge going septic, that could be from dead zones in the treatment process where sludge is again treated, or it could be toxic loads to the system. Those septic loads, like we said, can be from dead spots in the collection system, or if your lift pumps aren't timed correctly, or kind of similar situations with too much sitting water. Now, toxic loads are usually due to unusual chemicals from commercial or domestic sources. Like I said, it can either change the smell of your facility, your activated sludge, or it could discolor it. You could see, your sludge darkened increasingly. So, it could be black or it could be gray. You can see it, be shocked, so it's white. If that kind of changes from that deep brown color or kind of slightly grayish color that we're looking for, that may indicate, some toxic shock. So that's a good visual indicator of when you might have an issue. And then another way that we can kind of visually see issues with our wastewater is we see things like poor settling, which is commonly called pin flock. Well, one type of poor settling is called pin flock. And that is often due to a low F to M ratio or a long sludge age. This can be addressed by either subsidizing an increased BOD if your BOD is low with an additional food source such as methanol or dog food. Those are common sources of extra organic strength in your wastewater that some people use to subsidize. Or you can also address old sludge age by wasting more to decrease the microorganism concentration and decrease the sludge age. So this is what some fresh, this is what a wastewater facility that's running correctly, that activated sludge would look like, if it's healthy and it's processing wastewater correctly, it's treating organics. This would probably be removing ammonia efficiently. As you can see, like I said, it has that dark brown color, lots of little tiny white bubbles. So that kind of indicates that your oxygen is your dissolved oxygen is being delivered correctly. And then you don't see large bubbles large bubbling anywhere that indicates any line breaks. So when you're looking at activated sludge, it may look similar to this, but like I said, all plants can be a little different and it may look a little different based on your oxygen, but this is kind of what you might be looking for during that aeration phase for your activated sludge. So with that is the end of our kind of basics section of that. We have a quick poll question. Valeria, if you don't mind uploading that. So the first poll question we have is healthy activated sludge should have what kind of odor or smell? And the options that we have are fresh, earthy soil, no odor, rotten eggs, or bleached swimming pool. So let's go ahead and take about 30 seconds to try to answer that, and then we'll get going again. All right, that's about 30 seconds. Valeria, if you wouldn't mind ending the poll. We'll go ahead and share those results. It looked like we had 3 answers of rotten eggs and 14 answers of fresh, earthy soil. So most of us did get it right. It should smell like fresh, earthy soil. Sometimes it is odorous, no odor, depending on what it's like, but that may mean a lack of biological activity. Then, like we said, rotten eggs may indicate that there's something wrong with our activated sludge process. And if it smells like a heavy bleach smell, that's more of something that you'd want in your disinfection. All right, good job, everybody. Okay, so that brings us to our next section. So in this section, we'll kind of go more into detail about the characteristics and variables and how to troubleshoot some of those and what they mean, what they indicate in our activated sludge process. So we already talked about influence, kind of some of the general overview stuff that we had kind of talked about last webinar as well, with understanding our solids content and our organics content. But I do want to go into a little more about the influent flow rate and talk about how it can affect the activated sludge process. So the influent flow rate determines the time wastewater spends in our treatment system. This is known as our hydraulic residence time. Now, the hydraulic residence time of the activated process will change by both increasing and decreasing influent rates. A higher influent flow rate will mean a shorter hydraulic residence time, and a lower flow rate will result in a longer hydraulic residence time. Now, the hydraulic residence time influences the effectiveness of the biological treatment by providing microorganisms enough time to degrade the organic matter. A longer hydraulic residence time can lead to better treatment results by allowing microorganisms more time for the biodegradation of pollutants, but a low flow rate may lead to worse treatment results since wastewater may not be treated for the required amount of time. But then again, if it has too long of a hydraulic residence time, that may mean that you are using more energy on it, you are not optimized and stuff like that. So as with all processes in wastewater, you want to make sure that you have kind of balance through all of those things. So now we just kind of want to monitor our activated sludge process and our influent coming into our process so we can know if we need to make any adjustments. For example, well, and then we have a nice list here kind of showing, the unusual increases and decreases in our flow rate can impact some of the following characteristics, such as the hydraulic residence time, wastewater dilution, solid settling, oxygen transfer, shock loadings, and organic washout. So we want to be able to make sure we understand what's coming in and how fast it's coming in and how to adjust. For example, if we get surges in our wastewater due to infiltration of rain, this may lead to stuff like organic washout, where the microorganisms in our facility are moved too quickly out of the process, and we aren't able to properly treat all the organics in our wastewater. So, we want to understand if we have increased wastewater, how can we adjust? We would try to, slow down our detention time in other processes, possibly. If we have a redundancy or an equalization tank, we can maybe store some of that water temporarily. We may increase oxygen so we can kind of increase the rate at which we're treating wastewater and stuff like that. So those are a couple of solutions when we have an increased wastewater. And then when we have a decreased rate, in order not to dilute too much, we may increase like recycling. We may increase our detention time and our clarifiers, as long as we're not kind of exceeding those nitrates or something like that. But yeah, so that's why we want to make sure to understand our influent flow rates and how to make adjustments to them. Now, when we're troubleshooting, like we said, so if we have some increased flow rates, something we may want to do is increase our dissolved oxygen concentration in our activated sludge. This will make sure that we get efficient treatment. We may want to divert flow and treat some of that wastewater at a later time. Or we may increase our MLSS, or return activated sludge, return rate, to account for some of that washout that occurs with that increased hydraulic time. Now, when we have decreased flow rates, we may want to decrease our DO to kind of slow down the process and make sure that we're giving ourselves enough time, and we're not going to be getting rid of all our wastewater. We're not maintaining a micropopulation in our activated sludge. We may also decrease the MLSA, so return activated sludge, so there's less microbes in the activated sludge process itself. And because of that, there's less organics that need to be treated as well, and that will help decrease things like our solids that we are discharging as well. Now, we talked about last week about our TSS and our VSS. I just wanted to kind of show another slide on that, just to kind of show the kind of desired range of the effluent of our secondary clarifiers. So most of the time we have influent of about 150 to 400 milligrams per liter in our activated sludge. Then in our mixed liquor in the activated sludge process itself, when we're reintroducing some of that biomass, our suspended solids, the mixed liquor suspended solids, will be between about 1,000 and 3,000 milligrams per liter. And then the effluent from the actual secondary clarifier will be between about 1 and 20 milligrams per liter. So as you can tell, even though we have a somewhat high amount in the influent, and then we increase that to obtain treatment, by the time we're discharging from our settling process, we're down to almost nothing, 1 to 20 milligrams per liter. One thing I like to point about, or a couple things I like to point out about the TSS and VSS is that increased TSS and VSS can lead to issues such as bulking and foaming, reduce oxygen transfer, and decrease in overall treatment efficiency. So we'll make sure that we're kind of not exceeding those limits, or our bacteria will grow out of hand and kind of exceed what we needed for treatment. The way to basically control that is adequate primary clarification to make sure that we're not passing solids into the activated sludge process, but also our secondary clarifiers are used to decrease those suspended solids either through the wasting or the return activated sludge as well. Now, I'm not going to go too much into this again, but I just want to give you guys another slide as a reminder. So we have our activated sludge, the different types of bacteria. We have our nitrifying bacteria that use oxygen to remove ammonia. We have denitrifying bacteria, which are facultative anaerobes that remove nitrates in the absence of oxygen. We have our heterotrophic bacteria, which are our BOD consumers that use oxygen. And then we have our flock-forming bacteria, which will be those filamentous bacteria, which is important to this particular, some of the stuff we've talked about, because if we don't control our flock-forming bacteria, it can result in things like bulking and foaming. The other variable that we're looking to control in our wastewater process is our BOD and our COD. We kind of introduced this last week when we talked about our activated sludge process. And this is an important parameter that goes into the calculation of our food to microbe ratio. So this helps us control our process and making sure that we have the right ratio of our organics that are coming in versus our biomass. And we want to make sure that we're constantly trying to make adjustments to account for any increases or decreases in our BOD. But it should be known that I introduced this last week, that it does take 5 days to get BOD back. It can be difficult to make adjustments on the fly. And this is more of a parameter that we test for and report on, and kind of indicates our kind of general day-to-day practices and how they're working. Last week, we also talked about our ammonia and our total kilojoule nitrogen, that TKN. Now, the untreated ammonia and TKN, this is important because it can result in eutrophication in our receiving waters. This is excessive nutrients in those eutrophication is excessive nutrients in those receiving waters that produces excessive vegetative growth and depletes the oxygen in the water, and can result in the eventual die-off of aerobic microorganisms. So that's why we want to go ahead and some processes do treat for ammonia. So it should be, those treat processes should be aware of what's coming into the plant, how much they need to treat it, and then also how to troubleshoot elevated ammonia levels in your process. Typically, you want to increase your DO when you receive elevated ammonia, or you're seeing elevated ammonia levels in your discharge. But it can also be addressed by increasing recirculation or optimizing your pH and adding alkalinity. That's done because those bacteria that carry out the nitrification, they are very sensitive to pH changes. So you kind of want them in that optimal 6.5 to 8.5 range. By keeping it there, you can make sure that they're not shocked and that ammonia can be properly treated. Now, kind of hand in hand with that nitrate, or with that ammonia, is our nitrates. So as you treat ammonia, you produce nitrates. And the nitrate, as we mentioned last time, if you have excessive amounts of that in your drinking water, it can result in methemoglobinemia, which is that blue baby syndrome. In order to treat nitrate, you will typically introduce an anoxic zone. These are typically modified processes. Not all processes have these. But for modified processes, you may have an nitrate removal zone, which is an anoxic zone that will use where those facultative anaerobes will use, like we said, the oxygen bound to nitrogen in order to consume BOD. If you find that you do have excess nitrates, you can kind of increase the amount of areas that don't have oxygen in it, and then you can also increase your recycle rate to those anoxic areas. So the next control variable that we want to talk about is kind of a combination of temperature, pH, and alkalinity. We've kind of touched on these a little bit, but they're important because increased temperatures can result in increased microbial activity, increased nitrification. But if our bacteria have too much, if the conditions are kind of too advantageous to them, that can result in excessive growth and bulking or foaming. Now, our low temperatures tend to decrease our BOD removal, increase denitrification, and can increase our oxygen solubility, which means that you don't have to use as much oxygen in them. So it's good to kind of understand what those mean when they're kind of occurring. Our biological interactivated sludge process prefers to operate in that 68 to 95 degrees Fahrenheit range. But either way, temperature changes excessively can kill microbes and inhibit our treatment process. And then, like I already kind of mentioned, we want to keep our pH kind of inside that 6.5 to 8.5 range. Sometimes you have to make adjustments such as adding a caustic substance to it to account for any like low pH and stuff like that. So the next variable that we'll talk about kind of troubleshooting is dissolved oxygen. If you remember from last time, dissolved oxygen is introduced to our activated sludge process by our blowers into the aeration tank, and typically is distributed to our activated sludge by diffusers. Now we want our DO kind of in that range of 0.5 to 4 milligrams per liter. But kind of as a reminder, the optimal range is more of 1 to 2 milligrams per liter. And any excess dissolved oxygen tends to not result in any increased treatment, but can require more energy to produce. Now, when it comes to this specific variable, there's a couple ways that we can or a couple issues that arise from inadequate oxygen. When you don't have enough dissolved oxygen in your process, you can have inadequate treatment. Well, inadequate aeration tends to lead to inadequate treatment of your overall activated sludge process. Higher temperatures can lead to dissolved oxygen issues because, like I said, oxygen is more soluble in colder temperatures. So when you have increased temperatures, this will inhibit the ability of your wastewater to hold on dissolved oxygen, and it's less available for your microbes. When you have those increased oxygen, you have too much production and growth in your wastewater. That can lead to clogging and fouling of your membranes. And then things like high organic loads, those excess microbes, excessive solids, and toxic shocks can also inhibit your ability for microbes to be able to, or for the activated sludge process to occur. So when you have unusual DO issues rise up in your activated sludge, it's good to be aware of what might be causing it and what can be some of those issues that occur through it, which is what we kind of want to show a list of here. The next variable I'll kind of mention just briefly because we went over this last week is the sludge in our secondary clarifier. This includes the return activated sludge. This is the sludge that's pumped back to the activated sludge process. It is kind of our cultivated microbes that return as our biomass to break down more wastewater. Then we have our waste activated sludge, which is how we control our sludge age and our food to microbe ratio. This is sludge that is removed from the activated sludge process. And both of these are examples of how to control the sludge blanket, the secondary clarifier, and indirectly the settable solids that we may be discharging from our secondary clarifiers. So those are kind of more in depth about a lot of the variables that we mentioned last week, and kind of what some of the results can be and how to control them a little bit. But we're going to go now more into our calculatable variables and talk about what they mean and what they kind of indicate and how we can kind of calculate some of this. So the first one, if you remember from a little while ago, I did talk about settability and our settability tests. And the way that we test for settability in activated sludge is going to be what's called a sludge volume index test. It's called a settability test, but what the result is the sludge volume index. So what this test comprises of is you take 1 to 2 liters of activated sludge sample, preferably 1 liter, and you put it in a large open mouth beaker. You let it settle for 30 minutes. You record the volume of the sludge, that brown part in this picture, at 5 and 30 minutes. You do the 5 to kind of understand if the sludge is settling too quickly, and then the 30 will give you your variable that you use in the equation. So this is what that equation looks like. We're not going to calculate any of these equations in this class, but we will have some math classes later down the line to go more in depth of that. But this is what that equation looks like, and that gives us an idea of how quickly our sludge is settling. So that SVI, an example of a good SVI, a healthy SVI for your activated sludge would be between 80 and 120. And then the units they usually use is milliliters per gram. So 80 to 120 milliliters per gram indicates good settling. Then an SVI of less than 80 tends to indicate an old sludge age. And then an SVI of greater than 150 typically indicates filamentous issues. Now, if you have old sludge, that typically means that you need to waste more. If you have that filamentous issues, that may mean that you are providing too much oxygen, that you need to increase wasting as well, and stuff like that. So increasing the wasting will decrease the activated sludge process's sludge age and should increase the SVI. And increasing the return activated sludge will return biomass to the aeration basin and decrease the SVI as the average age of the sludge age is increased. So if you have too low of an SVI, you can do that as well. Now, this is also a useful test for some of those visual observations that we mentioned. As you can see on the left, there's a lot of particles in there. This may be either bad settled sludge or maybe earlier in the SVI test. But as you can see on the right, you have a lot of clear supernatant between them, and that kind of shows good settling. So if this is kind of a good snapshot of how our settling tanks would perform and give us an idea if we are settling correctly as well. This is that kind of step where you can look for those unusual settling characteristics like pin flock. Now, the next component, kind of related, is that mixed liquor suspended solids, the MLSS, or the mixed liquor volatile suspended solids. I went into detail about how to analyze for this in the last training. It's kind of similar to a TSS test, or you use a probe. But I do want to mention that we use this in our other calculations, such as our F to M ratio and our sludge age calculations. And then the normal operating parameters tend to be between 1,000 and 4,000 milligrams per liter. And then in terms of control of our MLSS, you can decrease your MLSS by increasing sludge waste, and you can decrease MLSS. I'm sorry, I had that backwards. You can increase your MLSS by decreasing your sludge wasting, and you can decrease your MLSS by increasing your sludge wasting. So it's kind of inversely related to how much you're wasting. Um, and then if you'll remember, the volatile indicates that living portion of MLSS, and usually is between about 70 and 80% of the MLSS. So that brings me to the second calculatable variable that we're going to talk about, that F to M ratio. This is our food to microbe ratio. So this represents the amount of organic material entering a bioreactor compared to the mass of microorganisms available to consume that organic matter. Um, the BOD is going to be the representation of the food that's in the bioreactor, and our MLVSS will be the part of the equation that indicates the microbe. So monitoring our FTM ratio is essential to ensure that the bioreactor is treating wastewater correctly and our permit requirements are being met. Our low FTM ratio will tend to indicate that our microbe populations, we may have too much biomass and not enough food. You may want to decrease your wasting. And a high FTM ratio may lead to excessive sludge production and overgrowth due to those high amounts of organics, and also an increased threat that we may be putting excessive nutrients in our wastewater discharge. And then the last thing before we move on from this slide, explaining FTM ratio, is that the FTM ratio tends to inversely correlate with our sludge age, that SRT or MCRT. We usually want our FTM ratio to be in that desired range of 0.2 to 0.6. And that's because, a low FTM ratio tends to correlate with high efficiency organic matter removal and longer sludge age. And then that kind of higher FTM tends to indicate that we may be having excess microorganisms, and we may be seeing an increase in microorganisms that we don't want to see in our wastewater. And again, to decrease your F to M, you may want to increase wasting and increase your DO. And if you have a high FTM, you may want to, if you have a high FTM, you want to kind of increase your FTM, you may decrease your sludge wasting, decrease DO, and add a supplemental carbon source if you have too low of DO, FTM ratio. Now, in order to calculate your FTM ratio, this is the equation we use. So like I said, that rate of applied food is going to be your biochemical oxygen demand, and the M is going to be your microbial biomass. In order to calculate that FTM ratio, you also need to know the flow going through the plant and the volume of your aerator. And you would divide the product of the flow times the BOD divided by the volume of the basin, of the product of the volume of the basin, and the MLVSS. So that's kind of just a quick kind of introduction of how to calculate that. And then, like I said, we also have the sludge age parameter. Our sludge age will indicate the average time that microorganisms or solids spend in the activated sludge process. Kind of on the general level, this is explained by the mass of solids overall in the aeration tank, and the mass of solids kind of entering the aeration tank. That kind of gives you an idea of the retention time of those solids, and more specifically the microbes. So that's kind of like the general way of calculating for just general sludge age. But the way we tend to calculate this in our plants or in the certification tests is by using more specific versions, such as the solids retention time, which is the SRT, or the minimum cell retention time. Now, these are pretty similar. A lot of the tests will kind of use these interchangeably. But in general, what they do is they represent the average time solids or a representative cell would be retained in the active sludge process, like the kind of average cell. And these are important because they directly relate to the microbial activity, the nutrient removal, the sludge production, the effluent quality, and then the energy efficiency. So if you have too young sludge, it's not going to remove nutrients correctly. There's not going to be enough microbial activity. And if you have too old sludge, you start to run into the same problems. And then also with the added thing is if it's too old or too young, it also affects your settling. So that's why SRT and MCRT are another parameter that you kind of calculate along with your F-time ratio to make sure that your process is optimized. And if one or the other or both is off, that kind of means, okay, well, if you have a high sludge age and a low F-to-M ratio, and your process does not seem to be removing enough BOD, enough ammonia, the solids are too high, that may indicate that you need to increase wasting. That's why it's important to remember that they're correlated. And if you have one high and the other's high, it either means that something's really odd on your process, or you may be calculating them incorrectly. But for conventional plats, you usually want to see about 3.5 to 10 days for an SRT calculation. And then, like I said, it is primarily controlled through wasting. And just a little more detail about the SRT and MCRT. I'm going to introduce the calculations. Like I already did mention that they are interchangeable. The stack state 8 courses tend to use the SRT, whereas like the ABC testing formulas and conversion table treats them both interchangeably. They use the same equation. Now, more specifically, the SRT is the length of time solids spent in the activated sludge and how long they've been retained in there. With the MCRT equations, sometimes do not include TSS and are only meant to account for the living solids. So this is what that typical SRT equation might look at. Like I said, sometimes this may say SRT, but typically it's trying to give you the solids that are in the activated sludge, the aeration basin, what's in the secondary clarifier, and that is divided by what is being wasted and what is leaving the plant as suspended solids. And you maybe, this is kind of what it looks like in more of a metric system, kilograms as opposed to pounds. And then if we have kind of more of an activated sludge process that doesn't include nitrogen, you may see an SRT range of 1 to 5 days. And that one brings us to how to calculate sludge wasting. So as I've mentioned, sludge wasting is how we kind of control the solids in our system. It's the process by which excess biomass or MLSS is removed from the activated sludge process. We usually refer to it as WAS, the waste activated sludge. And it is a variable that can be manipulated most readily to adjust that F-to-M ratio and sludge age like we mentioned. And that's because there is typically a limit to how much we can control of our solids in our influent, how much solids are in our effluent, and the associated biomass, or even the MLSS once the processes are ongoing. But if we remove them at the end by pumping them from the secondary clarifier, then we can adjust our solids, what we would call like our inventory in the activated sludge process. And then it is pretty simple to take that SRT equation that we showed you guys and rearrange it to calculate our ideal wasting rate. So this would look something like this. You take your solids that are in the aerator and secondary clarifier, you divide it by your target MCRT, then you subtract the total solids in your effluent, total solids in the flow, and you divide those by, or you multiply those and subtract from it, and then that whole thing is divided by the total solids in your wasting, the concentration. Yeah, and so that's kind of how you calculate that. Like again, we'll show you guys how to do this in another presentation, but that's kind of on the basis how that works. So that's the end of sludge wasting. With that, we have a quick poll question. We have 2 of them actually. Valerie, if you wouldn't mind bringing that up. So the first is, what is a good measurement of SBI for activated sludge? The answers are greater than 150, 0.2 to 0.6, 80 to 120, or less than 80. If you guys want to go ahead and take about 30 seconds, we'll go ahead and close that up. Or we'll go ahead and let you guys know what the answer is. All right, that's about 30 seconds. Valerie, if you don't mind closing the poll, sharing the results. So most of us got this right. A good SVI is about 80 to 100 and 120. We had a couple people indicate greater than 150. This would indicate that we might have more filamentous bacteria. If you'll remember, 0.2 to 0.6 was kind of that ideal F to M ratio. And then if you have SVI, no one answered this, but if you have an SVI of less than 80, that may indicate that you have old sludge. So it's time to kind of waste some and kind of change your activated sludge population. And then we do have right away a third poll question. If you wouldn't mind bringing that up, Valeria. We got other people. So this one is, what is the most effective way to increase the sludge age of our biomass? So if we want to increase the sludge age, would we add sodium hypochlorite? Would we decrease our aeration? Would we decrease wasting? Or would we increase wasting? Go ahead and start those, try to put in those answers and see how we do. All right, so we had about 15 of us answer, and most of us got this one correct as well. We want to decrease our wasting. So if you want to increase the sludge age, you need more solids, you need them to spend more time in there. So you would decrease the rate at which you're removing those solids. So you would decrease wasting. Increased wasting would do the opposite. That would decrease your sludge age. And then decreased aeration may not have a whole lot of effect on sludge age, but it would kind of change the amount of dissolved oxygen you're putting, and it may change your treatment efficiency. And then adding sodium hypochlorite may actually decrease your sludge age a little bit because you're probably killing off some bacteria. All right, good job, everyone. All right, so that brings us to our last little section. Here we're going to go ahead and talk about some abnormal operating conditions in activated sludge and how those kind of variables may lead to some of this. The first abnormal function I do want to introduce is mechanical malfunction. So we've talked about issues with equipment in other webinars, and our activated sludge is no different. So one of the most common reasons for abnormal conditions for our activated sludge process is due to mechanical, electrical, or control equipment failure. This can be partial or full failure, but either way, it tends to inhibit the ability of an operator to use activated sludge to treat our wastewater. Automation is an important factor in our mechanical malfunction, as automation does make our jobs easier, but the more mechanical and electrical parts we have in our system, that means there's more opportunities for things to fail and cause gaps in our treatment. Now, these types of issues are avoided by performing consistent preventative maintenance schedules, make sure that we have adequate reactive maintenance abilities, which can include proper maintenance knowledge and capacity, proximity to maintenance personnel with plant-specific capabilities, and the availability of redundant equipment. And then the last mitigation strategy would be to make sure to have reliable capital planning that ensures the funds are available when they are needed for repairs. Okay, so let's go into more of some of the biological abnormal conditions now. So a common issue that we tend to see is sludge bulking. This refers to the abnormal growth of microorganisms, specifically our filamentous bacteria, in the activated sludge flock. This causes the flock to become bulky, inefficient, and difficult to settle. This can also result in poor wastewater treatment performance, such as reduced removal of organic matter, nutrient, and suspended solids. Now, in order to control this, we want to make sure that we're monitoring and performing our process controls. We're identifying and reducing our filamentous bacteria. So when we observe bulking, we're taking samples and observing it under the microscope. Nutrient imbalancing. So if we have too much nitrogen phosphorus, we can lead to bulking. And then our sludge aging. So if our sludge age is, sludge age can be adjusted to favor the growth of flock-forming organisms over the filamentous bacteria, typically by increasing our sludge age. This can help control our bulking. And then also there's the chemical route where you kind of introduce some chlorination to damage the filamentous bacteria over our flock-forming bacteria. Another abnormal condition that is similar to bulking is foaming. So foaming is the formation of excessive foam on the surface of our aeration tank, secondary clarifiers, and other treatment processes. Similar to bulking, foaming is caused by excessive filamentous bacteria. The most common one we see tend to be the Nocardia and Microthrix parvicella, and then other surfactants, which can reduce surface tension and encourage foaming in our wastewater. Most of the foaming is categorized into 3 variations. It tends to be stiff white foam, brown foam, and very dark foam. There are a couple reasons for each of these types of foaming, but usually it's due to different types of microbes and nutrient imbalances that they result from. And then foam can also be persistent or unstable. Persistent foam does need to be addressed at the control level, and you need to make sure that you are balancing your nutrients, you have proper MLS, aeration, and then our unstable kind of out of the blue type of foaming usually can be addressed with some chemical agents such as chlorination or even mechanical removal such as scrapers. And then if you have too much surfactants in the wastewater, a lot of times you want to address that by community outreach and kind of talking to the industries that may be discharging to your wastewater facility. These kinds of issues, foaming is especially notable as occurring during kind of our seasonal changes when there's kind of a flip in the weather, kind of does something unusual to our biology and our activated sludge. And that's kind of the time when you want to keep an eye out for the most. Now, as we've kind of insinuated, sludge overproduction is a serious issue that can lead to abnormal wastewater conditions in our activated sludge. A lot of times, sludge overproduction can lead to bulking, increased sludge age, additional disposal costs, and reduced water quality. Some way to control this are maintaining that proper sludge age, the SRT, making sure that we don't have excess MLSS, make sure that we don't have excess nutrients, and that we're removing biological oxygen demand and to a lesser extent ammonia, and then make sure that we are not excessively using aeration. Now, the next abnormal condition, which is kind of related to a lot of those other ones, is nutrient imbalance. As we mentioned, kind of an unequal ratio of excess nutrients, such as nitrogen and phosphorus, can lead to the formation of bacteria that leads to those other conditions that we talked about, like bulking, sludge reduction, bacteria that overconsume DO, stuff like that. So in order to control this, we want to make sure that we are controlling our nutrients and kind of maintaining a ratio of nitrogen to phosphorus of about 5-to-1 or 10-to-1. Sometimes you may need to dose with excess nutrients to adjust if you are starting to see some of those results of the nutrient imbalances, such as bulking. A lot of times people add things like ammonia or urea or phosphoric acid to control your nutrient imbalances. Then you can also make adjustments to that by making sure that there's enough carbon that you're for your microbes to break down everything else, and that you are controlling your wasting and your return sludge correctly. The next one is poor flocculation. This tends to be the inability of microorganisms to form well-defined and settable flock structures. This will lead to difficult in solids liquid separation. So you'll have more suspended solids in your discharge. It increases your effluent turbidity and reduces your overall treatment. Older sludge tends to treat wastewater less effectively. So if you are seeing kind of, like we talked about in that settability test, you're seeing a very low SVI, and then you're starting to see an increase in your nutrients and your BOD and stuff like that. You want to increase your wasting to kind of decrease that and kind of increase your settling and your flocculation abilities of your biomass. Another part of this is make sure that you have proper mixing to make sure that organisms have the right opportunity to interact and create flocks. So here we're kind of just showing an example of some mixing, kind of showing how to make sure that our organisms and our biomass and our MLSS is always kind of being mixed correctly and what that should look like to make sure that they're able to form flocks correctly and that there is those interactions between the nutrients and our biomass are being carried out correctly. And then in this example, we'd also probably have decent nitrate removal if that is one of the permit requirements of our plant. And the last abnormal issue would be toxic loading. Like I said, this tends to come from industrial sources, but it also can come from domestic and some of our smaller commercial sources. Toxic loading is usually identified by odor issues, lack of treatment, sludge discoloration, such as white sludge or green colored sludge or very dark black sludge. And then, like, you know, if you do a microscope test, you can see your microbial activity dying off and being restricted. The best way to control toxic loading is to have the ability to have like equalization tanks or to be able to divert flow when needed, when you kind of see these spikes in unusual chemicals or if an industry notifies you. And then also having a good outreach and educational program that industrial, the kind of they use these industrial pre-treatment principles to make sure industries know what to do when they have when they accidentally discharge an illicit chemical or something like that. So that is the kind of last abnormal condition and the end of our presentation. We have one more poll question. Thank you, Valeria. So this, the last question is, which of the following types of bacteria is responsible for excess foaming and activated sludge? Now, this isn't the only one, but it's one of the more common ones. So the possible answers are Nitrobacter, coliform, archaean, and nocardia. All right, so that's about 30 seconds. So most of us did get this right. We got about 65% said nocardia. If you remember, it's the nocardia and the Mithrothix bacteria that tend to cause the foaming. Our nitrobacters, which is the second most common answer, those are actually participating in the nitrification process. Coliform bacteria tend to be an indicator organism, indicating that there's not enough that our wastewater may be infected. And then archaeans tend to be a different type of microorganism that produce methane. So good job, everybody. And that does bring us to the end of our presentation. Um, Thank you all for joining us. Is there any questions out there about activated sludge or troubleshooting? Um, we'd be happy to discuss them real quick. All right. Well, I'm not seeing any questions. I'll stay on for about another minute or two, but here's my contact information. If anyone has any questions, feel free to reach out to us if you have any questions about troubleshooting or any of the topics that we've covered. Um, and with that, we appreciate all of you guys joining us today. Thanks for staying over a couple minutes to finish it, and everyone have a great day.